All right, welcome everybody to Standing for Truth. My name is Donny Budinsky, and I am your host for today's exciting program. On Standing for Truth, we defend the truth of biblical creation. We also host some really awesome guests here on the program. And today it is a privilege to have Dr. Peter Line with me for this important show. Peter's work has been a tremendous blessing to this ministry. We've completed a must watch series debunking human evolution and the fossil record. We've done that together, and you can find that series on the channel in the description box of this video and also in our website, on our website, standingfortruthministries.com, under Peter Line in the guests on Standing for Truth section. Now, today, Peter, a neuroscientist, is going to dismantle human evolution by looking at the amazingly designed human brain. And I am excited for what Peter has to present today. Now, before I get into the presentation, though, what I'd like to do is give my guest, Peter, a formal introduction for anybody who might be unfamiliar with who he is. Peter's undergraduate majors were in biophysics and instrumental science. After this, he completed master's and Ph.D. degrees in the area of neuroscience, which is going to be his focus today, focusing on brain electrophysiology, and spent over a decade involved in such research. Subsequently, he worked as a university lecturer, teaching in the area of anatomy and physiology for over a decade. Peter has been interested in the creation evolution issue ever since becoming a Christian, as evolution was a stumbling block to him believing God's word was true. He has had a particular interest in the so-called hominid fossils for many years and has published several articles on the subject. And with that, Peter, I want to hand it over to you. Firstly, it's great to have you back. I know how busy you are. And so I always appreciate the time that you offer us here on Standing for Truth. Peter, how you doing today?
Not too bad, thank you, Donny. Yeah.
All right. Well, I'm excited. We got a comprehensive presentation. I've titled this specifically, Neuroscientist Demonstrates Amazing Design. So, Peter, I am going to get your slides up here. And whenever you're ready, my brother, the floor is yours to present.
Oh, thanks a lot, Donny. Yeah, yeah. In this talk, I'm going to give a very selective tour of the human nervous system, and I say selective because the topic is so large that basically only a tiny fraction of the nervous system could be covered in the short time available. And I'll be dealing with the human nervous system as I know it best. And in terms of what it can do, it appears to be the most functionally complex. However, a lot of the arguments for the design of the human nervous system also apply to the nervous system of animals. Regarding the mind, I regard it as immaterial and distinct from the physical brain. I just want to make that clear. However, the two go together, and so when talking about the brain, it may also include the mind, depending on the context. I'll deal specifically more with the mind later in the presentation. Now, if you want powerful evidence for a designer existence, then you don't have to look further than inside your own head. Now, consider that the 3D world we experience is essentially a construct by our brains. Now, whilst the brain may not be that impressive to look at, which weighing about 1.4 kilo or 3 pounds, it's often described as the most complex arrangement of matter in the known universe, and with good reason. And it's not just creationists that say that. The cover of a book, when I was doing a bit of research for this talk, on, actually that's from Amazon, How Your Brain Works by a new scientist. So that's an evolutionist publication. It says how your brain works inside the most complicated object in the known universe. And so it makes They don't say this, but it makes machines created by man, as great as some of those accomplishments are, they seem trivial in comparison. However, the new scientist promo at Amazon also said the following: "You are your brain. Everything that makes you you and all your experiences in the world are somehow conjured up by 1.4 kilograms of grey matter inside your skull. That might seem impossible, but science had advanced so much that we now understand not just its structures and inner workings, but also how it can give rise to perception, consciousness, emotions, memories, intelligence, sleep, and more." When I read that, I thought, really, so the brain's mystery essentially solved. That has been news to me and probably a lot of other people. Believe me, if the mystery was solved, you would have heard about it. But I should give them the benefit of the doubt, as that may not have been the message a promo was trying to convey, maybe just bad wording. Um, Unfortunately, even though I've ordered the book online and from two places, I'm able to obtain a copy of it. However, there are other new scientific publications published around the same time, and the following is another new scientist publication, and here it says, um, and that is quite a powerhouse you have upstairs. With its neural connections numbering in the hundreds of trillions, the human brain is the most complex object in a known universe. It is also, perhaps not coincidentally, one of the most mysterious. So now the more reality is it's a very mysterious object, and hundreds of trillions of connections. So if you had 100 trillion connections, um, someone actually in that publication counted that you would actually, if you counted one per second, it'd take you 30 million years. So that's how many if you had 100 trillion connections, and it likely has more. Another book I did manage to get a hold of this one, and I did read it. It's Your Conscious Mind. So that's kind of associated with how your brain works, as you focus more on the mind and consciousness. Um, Here it says, "What is this strange mental world that seems so essential to being human? The conscious mind brings together sensations, perceptions, thoughts, and memories to generate the seamless movie of a person's life. It makes us aware of the world around us and our own self. How all this emerges from a kilogram of brain cells is one of the greatest unanswered questions." Hence, now we go from understanding it to being one of the greatest unanswered questions, and the latter is really the closer to the truth, or the truth, I'd say. Um, Now, whilst there is considerable understanding of how the brain works in terms of how nerve cells function, fire, propagate action potentials, that is, and how chemical synapses work, releasing neurotransmitters and things like that. That's really known as that's the easy problem. Okay, the so-called hard problem is a different matter altogether. All right. So, according to the this book, the by New Scientist. But while we can explain, for example, how our eyes inform our brains about the wavelength of light that relates to a color, this doesn't tell you anything about what it is like to see the color red. In this view, even understanding every detail about the brain's functioning doesn't help us understand consciousness, because it tells you nothing about what it is like to experience red. And then it tells you the hard problem. There are a lot of hard problems in the world, but only one of them gets to call itself the hard problem, and that is the problem of consciousness. How a kilogram or so of nerve cells conjures up the seamless kaleidoscope of sensations, thoughts, memories, and emotions that occupy every waking moment. So, hence, whilst we may have understanding of some of the easier neuroscience problems, that is no more reason to believe that the physical brain created itself by a process involving no plan, purpose, and intelligence than it is to believe, for example, because we understand how a car engine works, it must have created itself without plan, purpose, and intelligence. Now, in the according to the late Francis Crick, who collaborated with James Watson in the discovery of the molecular structure of DNA. He later in his career turned his attention to neuroscience. Um, he writes, the astonishing hypothesis is that you, your joys and your sorrows, your memories and your ambitions, your sense of personal identity and free will are in fact no more than the behavior of a vast assembly of nerve cells and their associated molecules. As Lewis Carroll's Alice might have phrased it, you are nothing but a pack of neurons. This hypothesis is so alien to the ideas of most people alive today that it can truly be called astonishing. Now, such statements are simply pledges of allegiance to materialism and or Darwinism, that is, based on a person's worldview or ideology, not on fact or scientific evidence. In fact, commenting on Crick's this comment on in the astonishing hypothesis was John Horgan, who was a senior writer at the Scientific American at the time, and I presume an evolutionist. So he writes, in a sense, Crick is right. We are nothing but a pack of neurons. At the same time, neuroscience has so far proved to be oddly unsatisfactory. Explaining the mind in terms of neurons has not yielded much more insight or benefit than explaining the mind in terms of quarks and electrons. There are many alternative reductionisms. We are nothing but a pack of idiosyncratic genes. We are nothing but a pack of adaptations sculpted by natural selection. We are nothing but a pack of computational devices dedicated to different tasks. We are nothing but a pack of sexual neuroses. These proclamations, like Crick's, are all defensible and they all inadequate. So it just tells us nothing. You might, if it's saying that we're a pack of neurons, you can say we're a pack of electrons or whatever. It's just meaningless. Um, and so basically, it's shot down by his own evolutionary colleagues, such statements. Now, according to international winning author James Lifano in his book Brain Electrical Activity and actually I've got the book here. I think why us? I think I might have got the title. Yeah, no, I've got the title wrong there. I've got. He writes anyhow. He writes the distinction between electrical activity of the material brain and non-material mind of thoughts and ideas as two quite different things might seem so self-evident as to be scarcely worth commenting on. But for the neuroscientist, the question of how the brain's electrical activity translate into thoughts and sensation was precisely what needed explaining, and their failure to do so has come to haunt them. So for everything that the decade of the brain undoubtedly achieved, nonetheless, as John Maddox, editor of Nature, would acknowledge at its close, we seem as far from understanding the brain as we were a century ago. Nobody understands how decisions are made or how imagination is set free. Okay, now talking about the decade of the brain, which was in the 1990s, I remembered I spent countless hours crunching and analyzing brain data, or electrical brain data from recordings of brain electric activity and producing a lot of graphs and pretty color maps of brain activity. However, there's a world difference between seeing what parts of the brain gets activated during a particular task and explaining how that brain activity links to or translates into cognitive processing. That is how we think with our mind. Commenting on the situation in a recent book is neuroscientist Joseph Green, which is a pseudonym. He writes, "A technical ability to record from a brain perturbed and genetically manipulated has outpaced our ability to understand the neural mechanisms it exploits to function. In other words, while new technologies have enabled us to operate on the brain and offer new areas of inquiry, they have yet to produce a mechanistic understanding of how the brain functions. Certainly, a number of spectacular outcomes have been obtained, but these technologies have not yet been followed by breakthroughs of comparable size in our fundamental understanding of how the brain functions. We refer to this gap between our technological ability to act on a brain and our understanding of its mechanisms as a knowledge gap. and show how it fuels the popular misconception that our understanding of the brain is more advanced than it is. He goes on to write, the overreaching posture of neuroscientists who, like their counterparts in several other scientific fields, tend to adopt materialistic monism and push for a unified view of the world that reduces the unexplained problems to simple statements. For example, we are nothing but our brains, in which neuroscience can explain everything. This posture, which, as we show, lacks a solid foundation of scientific proofs, is far too comprehensive for neuroscience. And he says, while some neuroscientists undoubtedly share their opinions regarding the philosophical significance of their discoveries, their endorsement of physicalist viewpoints, that is, we are nothing but our brain, should be interpreted as a personal statement rather than one based on scientific findings. Now, I'll deal more specifically with issues regarding the immaterial mind later, but let's first look at how the material brain. Okay. Now, every awake second, environmental signals of some kind or another, such as light, sound, touch, or pressure, temperature, and airborne molecules conveying odors, are converted into electrical currents of ions called action potentials by sensory receptors in the skin or special senses like olfaction, vision, and hearing. Now, the action potential is the result of basically sodium and potassium channels on the axon membrane opening and closing, causing depolarization that is more positive and inside than with respect to the outside, and repolarization where the inside goes more negative again with respect to the outside. So that is, as a nerve impulse travels down the axon, sodium and potassium voltage-gated ion channels open and close. At the beginning of the action potential, the sodium gates open and sodium moves into the axon. That's depolarization. Repolarization occurs when the potassium gates open and potassium moves outside the axon, whilst around the same time the sodium channels close. This creates a change in polarity between the outside and inside of the nerve cells axon, and but the polarity is very localized. And the action potential, as shown in the diagram up there, the arrows moves across. The action potential, the impulse, continually travel down the axon in one direction only, because until it reaches the axon terminal, it doesn't go backwards from its starting point at the axon hillock where it starts, because it's not excitable membrane there. The excitable membrane has sort of sodium and potassium voltage-gated channels. Now, these ionic currents, bearing no resemblance to the information they carry, travel into the brain via nerve fibers, cables, if you like, illustrated in the above diagram of vision. The two optic nerves, each, so I can get my pointer in here. Okay, so you got the optic nerves there, each containing roughly a million or so nerve fibers, become two optic tracts at the point here, the optic chism. From there on in, the optic tracts aren't kind of labeled in this diagram. So basically, this is where the two halves of the nerve fibers from each eye kind of cross over, and then they travel basically to the lateral geniculate nucleus of the thalamus here. Okay, and basically from there the information then gets relayed from the thalamus to the primary visual cortex, and so basically your left brain basically gets the right visual field, and the right-sided brain gets the left visual field. The above basically illustrates how intricate, yet also how orderly the brain's hardware is wired with nerve fibers, like an integrated circuit board, for want of a better analogy. Once received, the brain somehow decodes the information and processes it so the original signal can be presented to you, the ghost in the machine, or mind or soul, whatever you want to call it. Now, all this brain processing occurs automatically without you having to think about it. Rather, you can think about what you see or what you hear, smell, taste, or feel in the case of input to the brain from other sensory nerves. Now, so basically this allows a person or you to see the world in vivid color, hear the sound of a waterfall, experience the heat of a hot summer's day, feel the breeze impacting on the skin, smell and taste food, etc., experiencing all these sensory modalities simultaneously in an integrated way in essentially real time. I say essentially real time as there is some delay of action potential because I travel at a finite velocity. The maximum speed is about 120 meters per second. Now, no one knows how the brain does this, but it surely involves signal processing orders of magnitude beyond anything we got. Even as this could or is explained one day, it would be like explaining how a TV converts a cable or error signal to an image on a screen, albeit more complex. Amazing signal processing, to be sure, but it doesn't explain how you, the ghost in the machine or mind, is able to perceive, that is, to become aware of through the senses uh, of this signal and think about the image or smell or smell or sound or whatever. For example, why would you find the colors of a particular sunset beautiful? A TV cannot dwell on what it outputs, as it has no mind or thoughts. Is it really feasible for an unintelligent process to have done this? Create the brain's hardware, then somehow enable the hardware to do sophisticated signal processing, and then create a ghost in the machine, so to speak, that we are clueless about how it works. This being created without plan or purpose by a process as brainless as a bacteria. I'm tempted to say somebody have lost their minds. Pardon the pun. Okay, so let's just look at a bit more about the nervous system. Although essentially a continuum, for convenience, the nervous system is divided into parts, regions, and systems. The brain, or known as the encephalon and spinal cord, medulla spinalis, form the central nervous system, abbreviated CNS. Extending from the CNS are cranial and spinal nerves. Here, can I get my mouse? There we are. Okay, so you have these nerves extending here. They constitute the peripheral nervous system. Now, the classification of the nervous system is in many ways arbitrary. There's a kind of box diagram I've kind of drawn up. For example, some equate the telencephalon, the two cerebral hemispheres, in the diagram. Get my mouse across. Some equate the telencephalon, the two cerebral hemispheres, with the cerebrum, and classify the diencephalon apart from not part of the cerebrum. That's fine. It really doesn't matter that much. But you can see here you've got the let me get my mouse back again. So you've got the human nervous system. You can divide it into peripheral and central nervous system. So the peripheral nervous system can be divided into afferent sensory division, efferent motor division. So the afferent sensory division is incoming information from special sensory receptors like your eyes, your ears, your ear, and things like that, olfaction. You can have the serial sensory division. That's information coming in from basically your internal organs. Then you have somatic sensory division is basically coming in from the skin, temperature, touch, pain, and things like that, proprioception, then your muscles. And then you have peripheral nervous system, the visceral motor division. That's the autonomic nervous system here. You have three divisions: sympathetic, parasympathetic, and enteric. We'll just have a look at them in a second. And then you have the somatic motor division, which is the skeletal muscles. Okay. And then the central nervous system, we'll look at more at that later. Spinal cord, brain. Okay, brain can be divided into cerebrum, cerebellum, brainstem. Brainstem can be divided into midbrain, medulla, pons, and the cerebrum are divided into telencephalon, which is essentially the two hemispheres, and plus the diencephalon, which is the main structures are the thalamus and hypothalamus. All right, now let's go in here. All right. So the autonomic nervous system, which at the I won't so much talk about the enteric nervous system, but the autonomic nervous system consists of different divisions that basically um, carry out the motor commands, and sort of a lot of them are under basically self-regulating or under sort of uh, autonomous automatic control, basically. So they contain neurons that innervate, for example, the heart glands and the smooth muscles of internal organs and blood vessel. So it's as mentioned, it's divided into three divisions, subdivisions: sympathetic, parasympathetic, and enteric divisions. And so with the sympathetic divisions that you can see on the right diagram, it generally inhibits or opposes the physiological effects of the parasympathetic nervous system. It tends to reduce digestive secretion, speed up the heart rate, and dilate the pupils. Um, and the parasympathetic generally inhibits and opposes the effects of the physiological effects of the sympathetic system, stimulate digestive secretion, slow the heart rate, and constrict your pupils. And the enteric nervous system basically is a collection of neurons in your gastrointestinal tract, basically, and sort of known as the brain of the gut. And it can function independently of the central nervous system. Basically, it is influenced by the sympathetic and parasympathetic systems, but also has independent control. And here you can see more of the you can see the parasympathetic innervation on the left and sympathetic innervation on the right. So we're starting to you can see the complexity of everything is starting to I hope it's starting to dawn on you. And if you're not used to neuroanatomy or things like that, it seems that understandably a lot of this goes over the top. My aim is not that you have to understand or learn everything, because that's really hard in the first thing, but just remember, all this is supposed to be created by a process as brainless as a bacteria. So, I don't know how, kind of that doesn't make me feel really great if something, the most complex thing in the universe was created by something as brainless as a bacteria. But it would make me feel a lot better if it was created by a supernatural, super intelligent creator God. Then I feel a lot better that I don't understand everything, because I don't understand certainly everything. In fact, it's a it's a bit weird to think that this was created by a process involving no intelligence, plan, or purpose. Quite amazing that people can believe that. Now, many of the self-regulating autonomic functions of the autonomic nervous systems are controlled by nuclei in the brain cell in the brainstem, as well as the hypothalamus of the central nervous system. Now, nuclei are clusters of neurons in the central nervous system, which are outside the cerebral cortex of the cerebrum, or the cerebellar cortex of the cerebellum. In the peripheral nervous system, nuclei are called ganglia. So, for example, within the nucleus of the medulla, which is the sort of bottom part of the brainstem, there you have cardiac and respiratory reflex centers, which are involved in controlling things such as heart rate and blood pressure. These are cardiac centers, and basic respiratory movements, that is inspiration, expiration, reflexes so respiratory. This is a respiratory center. Now, further up, next up, you have in the brainstem, the pons, you have respiratory center, amnestic and pneumatic, which modify the respiratory centers in the medulla. So it all starts to get, the complexity of regulating this all starts to pile on. Remember, all this has to be programmed into your DNA. The brain has to be built, and then it has to function. So this, it's quite amazing, actually, very amazing. Now, these nuclei that these in the brainstem that I was just talking about are all part of what's called the reticular formation, a group of about 100 or so small, loosely associated nuclei spanning the central core of the brainstem from medulla to midbrain, as indicated in the diagram. So you can see here. So this is the green part here indicates all these loose nuclei here. So when I say nuclei, a nucleus or a singular nucleus, that the neurons or cells in there perform a similar function. So that's why so different nuclei may perform different functions, but all the neurons in the same nucleus generally perform similar functions, which is another design feature that things are grouped together like this. Just get back to my slide. Okay. Now, although poorly understood, these nuclei are involved in all sorts of functions, including motor control, sleep and alertness, pain modulation, etc. The reticular formation plays a fundamental role in arousal and consciousness, control of movement and sensation, and in regulating the serial, that is, body in internal body functions of organs and things like that. You can see some of the things there. I'm not going to go through and name them, but yeah, I think you'll get the idea. Now, it just shows you a kind of a block diagram of things that the reticular formation, or what's called the reticular activation system, is involved in. So basically, it brings sort of sensory message from the reticular formation are brought to the brain cerebral cortex and vice versa. So it plays a fundamental role in regulating arousal and sleep, that is wake transitions, and it projects to the thalamus, things like the interlaminar nuclei of the thalamus, which projects diffusely to the cerebral cortex in wide region. Of course, the thalamus also will get to projects very specifically to certain regions, and some nuclei in the reticular formation, like the lucus ceruleus, project directly to into the cerebral cortex, bypassing the thalamus. Now, injury to the reticular formation can result in a reversible coma. So the question is, how did all these nuclei centers or groups, some vital life, get organized without planning or design? But as opposed to the evolution mechanism of natural selection, sort of gradient mutation, what would be the waiting time to create all these nuclei and their target cells and organs, as well as their integration? Even starting with bacteria, I suggest the waiting time will be forever. Such questions are not dealt with by evolutionists, as I just assume it happened and then invent just the stories of how it happened. In a way, that is the story of evolution. One big assumption after another, whilst ignoring insurmountable probabilities. Okay. Now, one can ask similar questions about the organization of the hypothalamus and its many nuclei, which are shown in the diagram there. Now, the hypothalamus is the main control center of both the autonomic nervous system and the hormonal, that is the endocrine system, as well as the center for integration between the two. The hypothalamus controls hormone production, the body fruits control of secretion in the pituitary gland. It helps regulate temperature, metabolic rate, water balance, appetite, and influences emotional behavior. It regulates reproductive development and functioning. Okay. Now, the above shows the major hormones or factors in the hypothalamus. The oxytocin is missing in the left column. It should be there because it's kind of produced in the hypothalamus, and it's kind of released in the pituitary gland. So yeah, near the blood vessels there, and then into the systemic circulation. But you have a lot of hormones, the thyrotropin releasing hormone TRH from the hypothalamus that trigger. We will look at that in more detail. That triggers the release of thyroid stimulate hormone in the anterior pituitary gland. But I'm not going to go through and name it all. But you can see there's a lot of hormones. The thing is, if you go here, hang on, you can see these are the releasing hormones. So you got one hormone is released in hypothalamus just to go into the pituitary gland to cause the trigger release of another stimulate hormone, which then goes to a target cell, which then releases another more different hormones. So there's kind of a chain effect here. So how do these things all get set up? You know, this chain effect. And just have a look at it. Next slide. I'm kind of navigating between the cursor and my computer, so sometimes it gets a bit confusing. Now, the above shows the location of the target glands and effector of some of the major hormones released from the anterior pituitary gland. There's sort of anterior and there's a posterior pituitary gland known as adenohypophysis or neurohypophysis, respectively. Um, okay, now I think the next slide is one I want to get. Yeah, here. So I want to look at the feedback control of the endocrine secretion of the hypothalamus, and the above is an example of how the hypothalamus controls the secretion of hormones by negative feedback control. So, for example, in response to stress, the hypothalamus releases you can see in the diagram the corticotropin releasing hormone CRH. Uh, and basically it releases it into the anterior pituitary gland, which causes the release of the adrenocorticotropic hormone ACTH into the blood. Actually, this step isn't even shows. That's kind of more complex than in the diagram. So the ACTH and stimulates the generation of glucocorticoids, glucocortico, glucocorticoids, that is cortisol in humans. Some of these names are difficult to pronounce. It stimulates the generation of cortisol in the cortex of the adrenal gland, which sits on top of the kidneys, which are then released into the blood. Now, and this hormone is known for its anti-inflammatory and immunosuppressive actions, sort of weakens the immune system. Now, once the increased level of this glucocorticoid hormone is sensed by the hypothalamus via sensors in the walls of the blood vessels. Once it's these levels are increased levels sensed, production is then sort of decreased. And it's an example of negative feedback control. So you have sensors. I'm not sure how the sensors work, but they have to detect increasing levels of these hormones, and then they have to give that information basically back to the hypothalamus to stop basically release of the stimulating the CRH or whatever that was stimulating it on top here. So basically producing CRH so that the levels of cortisol goes back to normal. And another one is another classic negative feedback loop is relates to the thyroid gland. So here you have basically thyrotropin releasing hormone TRH from the hypothalamus triggers the release of thyroid stimulating hormones in the anterior pituitary gland. This triggers the release of thyroid hormones from the thyroid gland, that is T4, which is thyroxine, which contains four iodines, and T3 triiodothyronine, which contains three iodine ions from the thyroid follicle cells. And this has the effect as indicated in the diagrams. So when these T3 and T4 levels get too high, the negative feedback, again, I'm not sure how, stops the thyrotropin releasing hormone, stimulating hormones from basically stops these hormones from being released, from triggering basically more thyroid hormone production. So this will then reduce the levels of T3 and T4. By the way, By the way, that's a reason in terms of, because I've talked before and discussed I suppose the hominids about correctness and things like that. That's the reason that people develop a goiter. People get a goiter or during fetal development if there's lack of iodine. Like cretinism happens if there's lack of iodine, because these the thyroxine and triiodothyronine, or the thyroid hormones, basically need iodine to produce. If you haven't got iodine, these won't produce, and these hormones are vital for development and things like that. And it's also why that basically, if there's a radiation fallout, that tell you to, I think it's potassium iodide or whatever, to use that. So you kind of saturate your foreheads with non-radioactive iodine, so you don't get, again, this radioactive iodine incorporated into your thyroid hormones. So anyhow, concerning the feedback systems, is it logical to believe such an ingenious method of negative feedback control came about unintentionally by chance, by a process that couldn't care less? As evolution is not conscious, it doesn't give a hoot. Um, when you read some of the evolution literature, you would think evolution is some sort of God, such as a magic powers attributed to it. Remember, this is just one of many such feedback loops in operation, and one of many variables for the brain to control. At what point does selection from one variable interfere with the selection of another? How does something get selected for amidst the noise of so many variables in the brain has to deal with, not just hormones, there's a lot of other things. Again, what is the waiting time for all this information to be fixated in the genome? Now, I came across, I was actually reading this yesterday, I think, in the Neuroscientist Essential Guide, and it's talking about the developing brain before birth. And a new scientific publication, it says the growing brain is an extraordinary feat of evolutionary engineering. And it also says, if you consider the size of the construction job at hand, billions of brain cells and several million support cells in 4 major lobes of the cortex and tens of distinct subdivisions, it is a truly staggering feat of evolutionary engineering. Interesting that the word engineering is used, which is usually defined as the way that something has been designed and built, which usually implies there is an intelligent designer behind the engineering. To be sure, it is a great feat of engineering, but not by the brainless process of evolution, but by an intelligent designer, that is God. Now, a bit more about the central nervous system. There's about 86 billion neurons in the human brain, which is the current estimate, but it's hard to say how accurate it is. Used to be, you could say anything from 100 billions or trillion cells, but apparently someone found a way of counting it. I'm not sure how accurate it is. There's about 36 trillion cells in the body. There's as likely as many neuroglia supporting cells in the human brain as nerve cells. It used to say, up until recently, it used to be said that there are about an order of magnitude more neuroglia cells than nerve cells in the brain, but this figure is apparently wrong. So there's over 300,000 motor cells innervating skeletal muscle, several million sensory cells supplying information to the central nervous system. Each human optic nerve contains about 1 million or so nerve fibers, basically, which sends information to the thalamus and then to the visual cortex. An average nerve cell has about several thousand synapses, links with other nerve cells. Now, the brain is suspended inside the cranium and it floats in cerebrospinal fluid, known as CSF. The central nervous system it's heavily fortified, so it's basically protected by skull and the spinal column. It's also protected by a blood-brain barrier, which stops certain harmful substances or non-harmful passing into it from the rest of the body. So again, all these things are like the blood-brain barrier, things like that. They all have to be designed and built. I won't be dealing anymore with that bit. Now, up until recently, it used to be thought that there are more than 50 types of nerve cells, maybe 100 in the human brain. Not sure what the figure was for neuroglia cells. However, that number is now likely to be much more greater. It was just recently, in I think last year, that scientists at Cortical Institute, reading from the diagram on the screen, studied genes switched on in individual brain cells, a technique known as single-cell transcriptomics, revealing an astonishing diversity of cell types. We have more than 3,000 different kinds of brain cells. So basically, we had a single building of previous work mapping brain cell types in high resolution in single regions of the human cortex. The outermost shell of the brain, the newly published package, expands those studies to dozens, up to 100 regions across the entire brain. where the single region studies found over 100 different brain cell types. The new release data shows thousands of different kinds of brain cells across the brain. For many parts of the brain, that complexity and variety had never before been described. So this is an astonishing variety of number of brain cells. Where the neurons are neuroglyphs, I'm not sure what the ratio would be, to be honest, but it's an order of magnitude or so more than was previously known, and hence add an order of magnitude more to the number of variables in terms of brain cells that have to be taken into account when designing the brain. Is it really feasible that processes as brainless as our bacteria could engineer such information into the genome, no matter how much time is available? Okay, so just to go on a bit more about the central nervous system. So it consists of as mentioned, cerebrum, cerebellum, brainstem, and spinal cord. So the surface you got of the cerebral cortex, which is the outer grey matter of the brain, is divided into, as shown in the diagram there. You got the frontal, uh, the sort of reddish, pinkish thing, and you have the lobe, the parietal, the blue, green is the occipital, and temporal lobes is a yellow orangey thing, and at the back bottom is the cerebellum. Okay, and then you have the part of the brainstem, the pons and medulla. Okay, now there's also a lobe in the center. If you go to in the medial or midline of the between the hemispheres, you can see it. It's called the limbic lobe. Okay, in orange there. Now the cerebrum is the most largest and most prominent structure of the brain. It's divided into pairs cerebral hemispheres by the longitudinal fissure. You can see that. You can see it there as the thing spins around. And together, the two cerebral hemispheres are known as the telencephalon. That is the cerebrum minus the diencephalon. And you can see the diencephalon is just situated above the brainstem, as shown in the diagram there. As mentioned earlier, some equate the telencephalon with the cerebrum. That is, they classify the diencephalon apart from it. Anyhow, we thought about the diencephalon. The cerebrum is the most prominent structure in the brain, in the central nervous system. Now, the telencephalon differentiates into the hippocampus, involved in memory. It's an extension of the cerebral cortex, which has curled up the intermedial region of the temporal lobe. You can see the sort of red thing there. It consists of 3 distinct layers of cortex. As such, it is called neocortex, as opposed to the 6-layered cerebral cortex. Now, the hippocampus appears critical for consolidation of long-term memories, with the strengthening of synaptic connections between nerve cells thought to be how long-term memory is stored. Without the hippocampus, it's really, if that's destroyed, you won't form long-term memories. Now, long-term memory does not appear to be stored in any specific region, rather it's spread across the brain. How exactly the strengthening of synaptic connections allows information to be stored or encoded and later retrieved is a mystery. Now, the telencephalon also includes the basal ganglia, which is shown in kind of red in a diagram there, and consists of several large nuclei embedded deeply within the white matter of the cerebrum, and extends to the diencephalon and the midbrain. They're involved in regulation of the stereotype automatic muscle movements and a muscle tone. Okay, I'm going to try and go through a little bit quicker because for time's sake. Now, you have the corpus callosum shown in white in the center there. That's the great commissure of the brain. It allows the two hemispheres to communicate with one another. So it's a massive bundle of commissural fibers. The brainstem includes the midbrain, as mentioned, which in the diagram is the mesencephalon, the pons, and the medulla, which is in the diagram is labeled as myelencephalon, and medulla spinalis is a spinal cord. The hypophysis is the in the diagram, that's the pituitary gland. The epiphysis is the pineal gland. Neurons are shown in the superior and inferior colliculus, which is the posterior part of the midbrain. They're involved in saccadic eye movements and hearing, respectively. And the thalamus is part of the diencephalon, as is the hypothalamus. And you have the cerebellum that's involved in motor coordination. It's often called the mini brain, and it's involved in other functions too, like language. Also, the thalamus, we'll look at more detail, is also appears not just a relay station, but involved in other functions as well. So looking at the cerebellum, you have divided into 3 lobes. You have on the left there, you can see anterior lobe, posterior lobe, and flocculonodular lobe. If you stretch on the right, if you kind of stretch it all out, you can see how it kind of unfolds. The anterior lobe basically is important in control of limb and trunk movements. And by the way, the cerebellar cortex, in terms of controlling movement, they control on the ipsilateral same side, whereas the cerebral hemispheres, they control the contralateral side, the opposite side. So output from the cerebral cortex, which goes to the cerebellum, it goes via the pontine nuclei and the pons, has to be crossed back over. So that basically the information in the cerebellum is on the same side as the movement it controls or regulates. Now, the posterior lobe is important in movement planning and in non-motor functions of the cerebellum. And the flocculonodular lobe, it's located near the brainstem, the orange thing there. It's also known as the vestibulocerebellum. It's the only cortex in the cerebellum which has which outputs directly from the cerebellar cortex, say, to the outside of the cerebellum, like the brainstem, in this case, the vestibular nuclei of the brainstem. All other cerebellar cortex, they will relay the information through these nuclei deep in the center of the cerebellum before outputting from the cerebellum. So the cerebellum is an automatic processing center. So one of its main functions is with the regulation of movements by adjusting muscles, underlying posture, and equilibrium. So it maintains equilibrium by using information from the vestibular apparatus of the inner ear. So the flocculonodular lobe, which has a lot of connection with the vestibular nuclei, which has connections with the vestibular apparatus in the ear, is sort of heavily involved in that. Another main function is in programming and fine-tuning movements controlled at conscious and subconscious levels, such as learned movement patterns. Think of a gymnastic routine. So here shows the layers of the cerebellar cortex, not the cerebral cortex. So it's kind of free-layered. It has a large number of neurons, in particular Purkinje cells and granule cells. The granule cells are only excitatory interneurons in the cerebellum, that is, they have their cell bodies within the cerebellar cortex. There are billions of them. They're very small. And it's the cortex of the cerebellum is uniform without, consisting of 3 layers. The Purkinje cells, so there's an inner layer, and there's a middle layer, and there's an outer layer. So in the middle layer, you have the Purkinje cells, which are very large, and the dendritic trees, where they receive input, are the largest entire nervous system. They can receive up to input from about 200,000 synapses. Okay. The axons of Purkinje cells, they're the only axons that will emerge from the cerebellar cortex. And their axons are entirely inhibitory. When I say inhibitory, that means that whatever cell they synapse on, they're going to cause hyperpolarization, or cause the inside of it to get more to be more negative, so less likely to fire. And you have, as indicated in the bottom of the diagram, deep cerebellar nuclei. There's 4 bilaterally paired nuclei, which most of the output from the cerebellum has to go through before it goes out to other areas of the brain. And now the cerebellum is another universe of complexity that must be explained by natural selection, sorting, random mutations. All I can say is good luck with that. Now, the thalamus is a paired structure, so we're getting back out of the cerebellum into diencephalon. Is another pair. A lot of the structures in the brain, they're paired, and the thalamus is a paired structure. It's located in the center of the brain. It serves as a main relay station of information to the brain. All sensory pathways except for olfaction, the olfactory nerves travels directly into the telencephalon or cerebral hemispheres. They don't go through the thalamus, but all other inputs go via the thalamus, except there's one other one, as we'll get to later. There's a tiny suprachiasmatic nucleus in the hypothalamus near the optic chiasm, where the optic nerves cross over, which receives direct input from the retina's the ganglion nerve cells, basically via the retinohypothalamic tract, and that is involved in controlling melatonin secretion from the pineal gland. We'll get back to that. Now, motor pathways, for example, from the cerebellum and basal ganglia, also travel to the premotor and primary motor areas of the cerebral cortex via the thalamus. Now, based on their locality, there are 6 groups of nuclei in the thalamus, and some of these are further divided. I'm not going to go through and name and all. I'm going to try to move through some of this, but just giving you an idea of what it is, of the complexity of some of these things. So basically, you can divide the connections of the thalamic nuclei into sort of 2 major functional classes: is relay nuclei and diffuse projection nuclei. The relay nuclei project to specific regions of the cortex. For example, visual information from the retina is relayed to the primary visual cortex by the lateral genicular nucleus. The diffuse projecting nuclei, like the interlaminar nuclei, project to wide regions of the cortex, which are believed to be involved in arousal, that is being alert, awake, and attentive. Okay. Now, so according to Nicholas Sheridan and Prasanna Tati in a paper, the thalamus serves as the main relay station for the brain. Motor pathways, limbic pathways, and sensory pathways, besides olfaction, all pass through this central structure. The thalamus can divide into approximately 60 regions called nuclei. Each nucleus has unique pathways as inputs and various projections outputs, most of which send information to the cerebral cortex. Similarly, in a paper by Farmer and Sullivan, the thalamus comprises up to 60 cytoarchitectonically and functionally distinct nuclei, each of which has a different pattern of anatomical connections to subcortical, cortical, and cerebellar structures and regions. Traditionally, the thalamus was characterized as a central sensory and motor relay station of the brain, reciprocally communicating and relaying signals between subcortical and cerebellar regions of the cortex, playing a role in sleep, arousal, and primary sensory processing. Now, as discussed, I think, did I have a thing here? Yeah, okay. Before I get onto that, just getting my mouse back here. Now, as discussed above, the thalamus can be divided into 60 regions, approximately distinct region called nuclei, which has unique pathways and inputs and various projections and outputs, most of which send information to the cerebral cortex. Now, whilst being grouped into around 60 distinct regions, these regions of thalamus collectively receive input from numerous individual nerve fibers and send numerous individual nerve fiber outputs to other areas of the brain. Now, I don't have an exact figure. I tried to find it, but it's hard to find. But anyhow, to give you an idea, each human optic nerve contains roughly 1 million fibers, which are axons of the retinal ganglion cells, which make up the optic nerve of one retina. So they would go to the lateral geniculate nucleus of the thalamus, and this gets radiated to the primary visual cortex. So there'd be hundreds of thousands, if not millions, of fibers, I presume, going through the thalamus to the cortex in all these regions. Now, how in different regions, and you've got these 60 distinct groups of regions of information are being sent, relayed through the thalamus. How could this setup be designed without deliberate intelligence and engineering? Would you build computer hubs or telephone exchanges by potluck? Millions of wires being wrongly connected would be very bad for business. How could such an immensely functionally complex relay station and brain like the thalamus be thought of as having been built without intelligent design? It is illogical. Now, the Farmer and Solomon also say, similarly, the role of the thalamus in cognitive processes was historically considered to be passive, serving primarily as a triage center for information, determining what information will be passed on to higher cortical regions for processing. And I state also, over the past 2 decades, the characterization of the passive thalamus has been debunked by myriad studies, and the contribution of the thalamus to cognitive processes, including attention, speed of information processing, and memory, has become evident. So as more is learned, the role of the thalamus becomes more complex, and becomes more complex to explain by evolution. Okay, let's look at the grey and white matter of the cerebral cortex. So that the above shows a frontal or coronal section of a brain of the human brain, and so you have the outer core of the cerebrum is the grey matter shown there, and it's also known as the cerebral cortex, and it covers the surface of the cerebrum. The grey matter is a darker colored tissue and contains cell bodies and neurons, branching dendrites, and glial cells. They're not neurons, but they're the workhorses of the nervous system. So it tends to be 6-layered and is highly folded. And white matter, the inside there, is composed mainly of myelinated axons that speeds up the transmission of the signal, the myelin, and makes up most of the inner core of the cerebral hemispheres. So grey matter makes up about 40% of the cerebral hemispheres, whereas the other 60% is white matter. Now, gyri, basically the raised ridges, they're the folds in the cerebral cortex. Sulci are the kind of grooves in between the gyri, and if they're really deep, they're called the fissures or whatever. Like there's a bit of longitudinal fissure here. Here you see the corpus callosum. Now, so only about a third of the cerebral cortex is exposed on the surface. About 2/3 of the cortical surface is actually in the sulcal spaces. The buckling and folding in the cerebral cortex, creating gyri and sulci, allows a greater surface area of cortex to fit within the confined space of the cranial cavity without having to increase the thickness of the cortex. So again, that's an evidence of design, you know. But like everything, everything in the brain seems to have an order, and in the layers of the cortex, the 6 layers, again, it's order. So the 95% of the cerebral cortex contains 6 layers. It's called the neocortex, includes that which is visible on the external surface of the brain. You can show the 6 layers there. Sometimes these are subdivided into A's, and like 6A and 6B. You have the other 5%, it's known as allocortex. It's kind of morphologically distinct, usually buried deep within the temporal lobe, like the hippocampus, and the allocortex has variable number of layers, but always fewer than 6. Now, the cerebral cortex, the 6-layered cortex, usually varies in thickness from about 2 to 4 millimeters, being thickest in motor and association areas and thinnest in primary sensory areas. The thickness of the 6 layers of neocortex also varies, as does the density, size, and types of neurons within each layer. And you have all these, I'm not going to go through and discuss them all, but you can see the layer 1, 2, 3, the first layer sort of contains a few nerve cell bodies and things like that. It contains glial cells and things like that. The layers 2 and 3 increasing level of size, the size of the pyramidal cells increase. The layer 4 is the sort of tends to get a lot of input from the thalamus, so that's the input layer. So it doesn't have many pyramidal cells. The layer 5 is the contains large pyramidal cells. It's the sort of output, one of the main outputs from the cortex to the basal ganglia, brainstem, and spinal cord. Layer 6 is a fusiform layer, contains sort of spindle-shaped irregular pyramidal cells that project to the thalamus. And even the surface is topographically, you can divide it into regions as well, and it's divided based on sort of functions, and there's basically 50 to 100 separate areas, and the Brodmann's areas are probably one of the best classification schemes that's been around for a long time, and that was done about a century or so by the German anatomist Corbinian Brodmann. He identified over 50 divisions based only on neuronal architecture of the cortex, and research since then has confirmed or shown that different functional areas of the cortex have a different neuronal architecture. Now, then you can get to the spinal cord. That's also very organized. So basically, each spinal segment here. Let's see, can I get my mouse in here? Okay. So basically, you get incoming information from. They've got a spinal nerve here, which contains incoming and outgoing information. Then, as it gets near the spinal cord, it divides into dorsal roots, which is the incoming information, ventral roots, which is outgoing information. The dorsal root ganglions, that consists of nerve cell bodies, unipolar neurons, which exist outside the central nervous system. Okay, and basically you have the butterfly shape. That's the grey matter where all the nerve cell bodies are located in the spinal cord. That sort of configuration changes a little bit as you travel across the spinal cord. And then you have the white matter here, shown here as a posterior or dorsal white column. You have a lateral white column here, shown here, and then you have anterior or ventral white column here. Okay. Get back to my slide thing. I'll go to the next slide. Trying to get through this a little bit quicker because I realize it's having taught a lot of neuroanatomy. It's probably not the most exciting stuff, but again, what I'm trying to do convey to you is the complexity, but functional complexity, and the orderly arrangement of this. So if it goes over, if it's a bit too much, just think you're supposed to believe this was created by a brainless process. Now, the grey matter in the spinal cord, so you can see that that's in the center of the spinal cord. It's organized into divisions called laminar, often known as Rex laminar, 10 layers, divided on the basis of architecture and longitudinal organization. You can see they're numbered on the left 1 to 10, basically. 10 is the central region there. So the lamina run parallel to the long axis of the spinal cord. Now, these layers form part of either the dorsal or ventral grey horns, and sometimes lateral grey horns. Now, part of layer 7 is sometimes referred as a lateral grey horn, but it's only processed in the thoracic and lumbar segments of the spinal cord. Okay, so that's sort of more the central and low part, and they contain the serial motor nuclei. That is nuclei which innervate sort of internal organs and things like that. The dorsal grey horn is formed by laminar 1 to 6. So that's on the diagram, it's the upper end.
No, I think we might have lost Peter here. Let's make sure from the audience that's just not on my end. And let's see here. Okay. In the meantime, we will give Peter the opportunity to come back. Just 1 second. Okay, we'll give Peter the opportunity to make his way back in. Lorraine looks like has confirmed that he's frozen. Okay, well, firstly, we're at about the midpoint of Peter's technical presentation. Hopefully, he realizes that we've lost him as he is in his slideshow presentation. So hopefully, he makes his way back in soon. And so yes, he's frozen. We'll give him that opportunity. I do have the questions, the super chats, and all of that saved. I'm looking forward to seeing Dr. Peter Line engage those, as this is a comprehensive presentation and show that truly demonstrates amazing design and how the human brain refutes human evolution. And so a 21st century mind, no worries, my good man. He'll be back for sure. So, okay, let's go over some reminders and announcements. It's been an excellent week, as has been every week this summer, this epic summer. And so yesterday, I highly recommend people check this out if they have not yet seen it. It was a blessing and a privilege to have Jennifer Niza here from psychic to Christian. So we focus on everything supernatural, all things supernatural. This included demons, psychic mediums, truly undeniable evidence for the supernatural presented in this roughly 2-hour discussion and Q&A. So I do encourage both critics and non-critics to check it out. Any objective thinker watching this program, if they are a skeptic, they'll definitely go from skeptic to believer after watching this. Today, of course, Dr. Peter Line and his presentation on how the human brain, the amazingly designed human brain, refutes human evolution. And tomorrow, we'll be back here. This will be at 3 in the afternoon, so an earlier debate. We've had some earlier shows this week, just to kind of give everybody not only a diversity of topics to enjoy, but a variation in the times that we do our live streams, as I know people like to enjoy these programs live when they have the opportunity, when they have the time and chance, and interact that way. So tomorrow, the great Millennium Debate, Lucas Curcio and Keith Foskey. Lucas takes the premillennial position, and Keith Foskey holds to amillennialism. Both are experienced debaters. They both have studied this topic extensively. And so I think this will be a debate to remember on the topic of the millennium. For those that love a good old debate on eschatology, and specifically the millennium, the various positions on the millennium, post-millennialism, amillennialism, premillennialism. We've hosted a ton of debates on that topic. We kicked off this week on Monday with a debate between Dr. Sam Frost, who holds to amillennialism, and Dr. George Antonios, who holds to premillennialism, and they engage in this topic on Monday. So you can check that out on the channel. Now, discussing evolution and science-related topics. Last week, I was here with Dr. Jerry Bergman, a frequent guest and visiting scholar here on Standing for Truth, and he exposed the truth of Darwinism. This was a comprehensive program. We covered a lot of ground. We discussed vestigial organs and structures. We discussed so-called bad design, pseudogenes, mainly the vitamin C pseudogene, synthesis pseudogene, and many other topics. So this was a comprehensive one. If you have not yet seen it, please do make sure you check it out. It is up on the channel where you can find it. Now, pretty soon, we're going to be in August, and August is going to be another packed month of events. We want to give you guys the summer of all summers when it comes to interviews, discussions, debates, and more. And so we're going to have Dr. Jason Lyle here, Ph.D. young Earth creationist astronomer, and he is very well-rounded on all topics related to the origins debate. Truly an encyclopedia worth of information. And so Sam and I, we are going to be hosting this. Sam's going to be my co-host. We're going to be here for 2 hours live with Dr. Lyle. So this will be the critics' opportunity to join the live chat, present their objections, their challenges. Sam and I, we've been doing this long enough. We've hosted enough open mic debates where we know what the critics are going to say. We know their arguments. We know their challenges. And so we're going to present those to Dr. Lyle, and we're going to make this a comprehensive program. We are going to engage radiometric dating. We are going to dismantle the distant starlight challenge. Guys, this is going to be an excellent program. We are going to focus on the amazing evidence for young Earth creation. Josh C says Jason is awesome. Amen. I agree. So I'm excited for this program, specifically coming up in August. Other debates, notable debates here that we have in August coming up, the Great Purgatory debate. So we've hosted over 400 debates on all sorts of topics, or we're at least close to 400. And so this will be the first formal debate that we host on the topic of purgatory. And I'm exceptionally excited for this one because we have two experienced debaters, two excellent debaters, William Albrecht taking the Catholic position, Turretin fan taking the Protestant position. So it is a classic Catholic versus Protestant debate, and it's going to be on purgatory. We got another one in August that I just booked, another Catholic versus Protestant debate. That one will be Dr. Robertson Janis and Dan Chapa, and they're going to be debating the Eucharist. So this will be the Great Eucharist debate. So we've got some pretty epic debates for you in August. Here's another one, the deity of Jesus Christ. Sam Shamoon and Stacy Turbeville. Both of these gentlemen have had many debates in the past, and I know they are coming in swinging for this one. So this is going to be fun. This will be lively, I'm sure. It'll be very engaging and entertaining. So this is the Deity of Christ debate coming up in August. So I'm sure it'll be here in no time. More notable nature of God debates coming up. Albie Al and Taylor Stewart, the servant in Isaiah 53. So should be a good debate, interesting debate, an important one, that's for sure. So I'm excited for this one. We've got on the 31st. So we're going to end the month with a bang. Praise I Am and Caleb Percher. They are going to be debating the topic of the Holy Spirit and the Father. Are they different persons? Praise I Am takes the Trinitarian viewpoint, and Caleb Percher holds to monarchianism. And so this will be on August 31st. This is how we are going to wrap up the month of July. If you're not yet caught up on all the epic shows for July, do check the channel, as there is a lot for you to enjoy. Okay, here's a couple more debates. I'm excited for this one. Tristan Gabriel in the chat, 21st Century Mind. He will be here engaging Dr. Sam Frost. Dr. Sam Frost has been here several times in the past. And two true professionals here. They're going to be engaging the topic, the question, did the New Testament or did New Testament prophecy fail? And so this, if I remember correctly, in light of all the debates we've hosted, this will be the first time we host a debate on this specific topic. So I think this is going to be very interesting, and I am excited for that one. We've got a full preterism debate. So we've also hosted several debates on the topic of full preterism. This one here will be between two PhDs, two gentlemen who have studied this topic comprehensively. They have a lot of knowledge, and so they're going to give us a debate to remember, which is what we like here. So this will be Dr. Sam Frost and Dr. Jerry Woods debating the topic of full preterism. And you see in the chat, praise the I Am saying, yeah, buddy, on his debate coming up. Actually, he's going to be engaging in a 2-part debate series. So the first one is going to be at the end of July with Caleb Percher, and then Caleb Percher are doing a separate debate, and that one will be roughly a month apart. So that'll be in August. So Praise, I'm sure, is busy prepping for that one and getting ready to give us an engaging debate that we are all going to enjoy. We've even got debates scheduled out into October, September, October. So I'm currently in the works discussing details on some major debates coming up. This is one of them. This one is confirmed. This is a debate between Jake, the Muslim metaphysician, and William Albrecht. Was Justin Martyr a Trinitarian? So this one should be fun, should be an informative one. That's for sure, as they both have several months here to prepare for this debate. So guys, this is just an overall snapshot of what we have here on the channel for you. Coming up, I've been doing a series responding to Stated Clearly. This is John Perry from the Stated Clearly YouTube channel. Many in the camp of evolution, they argue that endogenous retroviruses, shared endogenous retroviral sequences in the biological world, they like to focus on the primates, is irrefutable evidence for evolution. Some say that it's as close to proof as you can get in science. As we know in science, you don't prove things. We have a hypothesis that we work to disprove. But the evolutionary community argues that ERVs, again, endogenous retroviruses, are as close to proof as you can get for something. So I've taken it upon myself to focus a lot of time, energy, and put in a lot of work to engaging that argument, as I like to focus on what the evolutionary community believes are the best lines of evidence for evolution, the best reasons why we are purportedly related to banana plants, whales, pine trees, and the great apes. And so I've been doing a series here responding to Stated Clearly's video, John Perry's video, on the 10 reasons why ERVs really are the ancient remnants of exogenous retroviruses, that they've essentially come from retroviruses. So I've done 2 episodes so far in that series. It is a technical series, but it's an important one, as I did not see anybody engaging John Perry's specific video there. And so I've wanted to take it apart point by point, argument by argument. And so you can see those videos on the channel as well. And part 3 will be focusing on the origin of retroviruses in general. And so I should be getting that out there, hopefully relatively soon, for people to enjoy. And I have not seen really any real good, solid rebuttal or counter to the arguments that I've made and presented in this series, but also in my book that I put out on the endogenous retrovirus argument. It's called the Endogenous Retrovirus Handbook. And so you can check that out. I've also got plenty of content here on other so-called lines of evidence that evolutionists argue provide irrefutable evidence for common descent, like the chromosome 2 fusion. So you can find lots of content on that as well on the channel. Specifically with Dr. Peter Line, he did a 3-part series with me, comprehensive series, refuting human evolution like he is today. Today he's doing it from a neuroscience perspective. But we did a 3-part series, comprehensive series, probably at least 9 or 10 hours worth of content, engaging human evolution from the fossil record. And so we focus on all your famous hominins, your icons of evolution like Lucy, Australopithecus afarensis, the Australopithecines in general. We did an entire show on the Hobbit and Naledi, and also engaged Erectus, Neanderthal, Heidelbergensis. So you could see that probably in the description box of this show here. 21st Century Mind says, is he coming back? I hope so. I've called him twice as I was going over reminders and announcements, and there was no answer. So my fear is that since he was in his PowerPoint presentation, he did not see that his internet cut out. And so he may have just continued on with his presentation. So I hope that's not the case. But when you're in PowerPoint and if your internet goes out, it's not really going to notify you unless you're in an internet bar. So we were supposed to take a little break midway through, an hour through, or into the presentation for some audience questions. So I'm hoping that he realizes soon that his internet went out. And so, yeah, hopefully he's coming back. He should be once he notices. I'll give him another call here because I really want him to engage these audience questions, unless we just end up making this a 2-part series if he doesn't make his way back. So, okay, let's get some questions for me, I guess. While we wait for Peter, I will engage the audience a little bit, give him some more time to hopefully make his way back in. Lorraine Drosophila, appreciate you being here, and always enjoy your questions and interaction in the chat. That's for sure. We love our critics, and we like to leave no stone unturned in these programs. And one way we can do that is by having our critics and having our favorite skeptics here to interact with. So Lorraine says, do you have evidence of a special ERV sequence in humans? Well, I discuss in my book on ERVs and also in my videos that we can begin to determine what is a created ERV sequence or unit, okay, and what is not. And to do so, we need to look at that which is fixed. Fixed just means in genetics, stuck in place. So if everybody has this ERV sequence or unit, because there's also what's called solo LTRs, okay? These are just functional stretches of DNA. They have important functional roles. They serve as binding sites for transcription. They can act as promoters. And so it's the evolutionary community that argues these solo LTRs are basically just degenerated ERVs. They were once ERVs, but now they've lost your key ERV components like your GAG, your POL, your ENV components or genes. And so that's just an evolutionary assumption. That's just an evolutionary presupposition. They have no evidence for that. What we see is a functional stretch of DNA, these thousands of solo LTRs. LTR stands for long terminal repeat, that are shared across the biological world. Okay, so we share thousands of those with the great ape, specifically the chimpanzee. We have no reason to believe that those solo LTRs were once full ERVs. Okay, your full ERV comprises what? LTR on either side, GAG, POL, and ENV components or genes, okay? Your ENV is the envelope protein. It stands for envelope protein of the ERV. Now, this ERV shares sequence similarity to exogenous retroviruses. An exogenous retrovirus is built the same. LTR, GAG, POL, ENV. Okay, so the evolutionary community argues that the ERV must have come from a retrovirus, an exogenous retrovirus. Exogenous occurring from outside, horizontal transmission. The HIV virus is a well-known or famous exogenous retrovirus. And then you've also got an ERV, an endogenous retrovirus, meaning it occurs from within. Okay, so the question is, are these really the ancient remnants of past viral infections, or are they created units of DNA function? Because if they are created units of DNA function, then we have an elegant explanation or answer to the question of why do we share these with other creatures in the biological world, like the chimpanzee, for example, our so-called closest cousin in the biological world in the evolutionary family tree. Well, the answer is simple. The same reason why all modes of transportation have engines in common. It's good design. They're there for a functional reason. They have a very significant purpose. Okay. And so the reason why ERVs share sequence similarity with exogenous retroviruses is because they have to in order to carry out a number of their functional and operational roles. A couple of those operational roles are involved in the innate immune system and cell stress response. They fight off harmful viruses. They act as antiviral protectors in the genome. They are these front-loaded antiviral programs, amazingly designed and sophisticated DNA units. And then they are also involved in what's called viral mimicry. And they do this by fighting off or destroying tumor cells. And they have to look like the exogenous retrovirus that they are mimicking in order to carry out those roles. Okay. Now, the so-called ERVs that don't necessarily resemble your retroviruses, the ones that you have just a solo LTR, okay, those are only assumed by the evolutionary community to be or to have once been an ERV. But no, what we see is they have a function. They're tightly integrated into biological systems and genetic systems of biological organisms, and they have important functional roles. And so the best explanation for those is that they are simply functional stretches of DNA. And so if they're fixed, if they're functional, if they're not disease causing for the most part, they are created. We have good evidence for that. Now, with Lorraine's, to answer her question in its entirety, ones that are not fixed, ones that are disease causing, and ones that are not functional, at least in terms of having an essential function, like in placental development and embryological development, those we would argue are from true infection, passed on vertically, hit in the right spot, and they've become endogenized. Okay. And so let's see here. Looking at the chat. Lorraine says, but function is not a refutation of common ancestry. If that function has no explanation in terms of how it could have originated due to selection, due to evolutionary pressures, so-called beneficial mutation accumulation, then yes, it is a refutation of common ancestry. These fixed ERVs are tightly integrated into these biological systems, these genetic systems, and they have functions that the evolutionary community simply don't have evidence for how they could have originated naturally. That's why my challenge to the evolutionary community has been and continues to be, show me an example of a non-functional endogenous retrovirus going from non-functional to something as functional as these ERVs that we see are involved in embryological development. They're involved in the placenta. And so you can find a number of papers in the conventional literature. One that specifically comes to mind literally admits, they confess that if we did not have these ERVs, then mammals as we know would not exist. I like to exist. Lorraine likes to exist. We like to exist. And we actually would not exist without these ERVs and ERV units. And so that tells us that they are essential. And so the evolutionary community, they have some nice philosophy. Okay, they have a great story. Evolutionists hope, dream, and imagine that they're related to whales and pine trees, but without any empirical evidence, then it's just pseudoscience. It's just a story. It's their philosophy. And so we need to see how these functional roles can come about. Okay, because if viral DNA randomly inserts itself into a cell, into a host cell, it's more likely to disrupt genetic functionality than make it better. It would be like if a painter who was blindfolded, if he were to apply random brush strokes to a painting he was working on, he's more likely to ruin the painting, to make it worse than make it better. Okay, and that's why it makes more sense that these ERVs with these essential functional roles were front-loaded in the beginning by God, by a designer. And so we share these ERV units with the biological world because it's good design. Humans build in homologous patterns. Humans build in nested hierarchical patterns. And so why shouldn't God? The Bible says that we are created in the image of God. And so we should be able to get a sense for how God created based on the way we create, based on the way we design things. And it just so turns out that humans build in homologous patterns. Humans build in nested hierarchical patterns. And so this just happened to be the case. And it turns out that God designed a biological world with similarities in anatomy, morphology, genetics, and physiology, and also built in hierarchical patterns. Every single creature has to be more similar to one creature and less similar to another. So, for example, humans were designed with RNA, with DNA, with proteins. So are other creatures in the biological world. Okay, we're all built with these basic building blocks and these information systems. Okay, highly complex information systems like DNA. And so the of course we're gonna share more with, let's say, the chimpanzee. We look a lot more like the chimpanzee than we do with a banana plant or a dog. And so there's this natural nested hierarchy where, yeah, humans look a lot more like the great apes than they do, or we do, with a banana plant or a dog. So it's just a natural nested hierarchy. A sedan has more in common, looks a lot more like a hatchback than it does with a van. So the hatchback and the sedan are going to share more with each other than they will with a semi-tractor trailer or an airplane or a boat or an unpowered vehicle like a bicycle. Okay, there's a natural nested hierarchy that exists in both the design world and the biological world, showing that the biological world is indeed designed by God. But God is such a more sophisticated designer than we are. We're just in the infancy of recognizing design. And so God was able to create his creation. He was able to design the biological world with the ability to reproduce, to pass on their genes. This is incredible design. This is incredibly sophisticated, and it defies naturalistic explanations. So, okay, let's see what else we have here. And I've given Dr. Peter Line now quite a bit of time, it seems, to make his way back in. So it's looking like we may have to turn this into a two-part show and note where he dropped out in his presentation. He dropped out about an hour into it. I'll save the questions, especially the super chats. 21st Century Mind, my good man. Excited for your debate coming up. He says, could you please answer my two questions? Since they are neuroscience related questions and Dr. Peter Line is the neuroscientist here, I will want to give you the best benefit in terms of the answers provided to your questions. So I'll save those for sure, especially because you sent them in as super chats, and we'll get Dr. Peter Line to address them rather than myself. And see, okay, let's see what else we have here. Lorraine says, yeah, we wouldn't be mammals. Maybe we would be marsupials. Here's the thing. There are different designs in terms of reproduction in the biological world, in the same time that we have a variation of designs in the design world. That's why you can get various kinds of engines when it comes to modes of transportation. You get a regular engine, you can get a diesel engine, you got electric cars now. Okay. So just because one vehicle is a diesel and another one has a regular engine, or you can get one of these Tesla electric vehicles. Okay. It doesn't matter that well. So the diesel ones must be the precursor to your regular engines, because look, we have cars that can run on diesel rather than a regular. We have ones that are electric rather than a regular engine. Okay. Or you have an all-wheel drive, rear-wheel drive, four-wheel drive. No, these are just variations in design. So this argument is so weak. I've seen it used by evolutionists like Mark Reed and Dr. Dan Stern Cardinal. No, it's very unsophisticated. It's not convincing. We have variation in the biological world in terms of design. Yeah, that's amazing. You want just the same design for everything? No, there is variation. There's beauty. There's a diversity in the biological world, in the same way that we have a variation of designs in human-made things like in vehicles. So yes, you have marsupials. Okay, sure, you have variations in the design world when it comes to reproduction. We have, fish, amphibians, reptiles. We have mammals. This doesn't prove that mammals evolved from primitive mammals or creatures that weren't mammals. So the evolution community is going to have to do a lot better than what they've done because what they'll do is point to non-mammals, okay, and then say, well, see, creatures can clearly live without a placenta, or they can live without the specific reproductive mode that mammals do. No, it just shows that there's variation in design, because all of these different designs, all of these different biological systems, they're all very sophisticated, they're highly complex, they're fully formed, and they're doing the job that they were meant to do. Just like we have variation in human design. So again, there in the technical literature, we find conventional scientists admitting that if we did not have ERVs, mammals as we know could not exist and would not exist. So it's a pretty easy answer when the evolutionary community, critics of special creation, say, well, why do we share ERV and ERV-like sequences with the biological world? Let's just focus on mammals. Why do all mammals have these ERV sequences in common? Very simply, it's good design. It's necessary. Literally, if we didn't have them, in the same way that if a car did not have an engine, it's not going to get very far. It's not going to be able to do what it was designed to do. And so these ERV sequences, again, ERV sequences function as antiviral protectors. They function in embryological development. They function in the gene expression process and pathway. They help to determine cell types. They fight off harmful viruses, including exogenous retroviruses. Okay. They function in viral mimicry. And so very sophisticated DNA units. Okay, as we are coming up at 8 and I have another appointment in about an hour, it looks like since we're coming up at the 2-hour mark here, we will have to turn this into a 2-part series with Dr. Peter Line. Now, Dr. Line has put a lot of work into his slideshow presentation. He's got about 95 different slides. And so today was going to be a 2-hour presentation and about 30 minutes of audience questions. And so I know he's got the slides ready. He's good to go with it. And so what we'll do is schedule a part 2 sooner rather than later. So next week's programs, we have shows on the 29th, the 31st. On the 29th, I'll be here with Dr. Dino. We'll be continuing our series responding to Crash Course's video titled Evolution, It's a Thing. Okay, so that'll be on, I think that's Wednesday. We've also got a show on the 31st. Oh, no, that's Monday, and then a show on the 31st. So we'll try and fit it into next week. Part 2, because I still don't see Peter and I don't see an email response from him. So praying everything's okay, but I'm suspecting his internet went out. It might have even come back on, but if the internet goes out even for a second, the feed drops and they'll have to rejoin. But if he was doing his presentation, he may have just went through and continued with his presentation. So I'll talk to Peter and we'll make sure to get our second part in here. And let's see. So young Earth creation has to show evidence of special creation. Yes. So I would recommend my 2 talks at the latest boot camp 2024. Matt and I did one, undeniable evidence for biblical creation and refuting the best evidence for evolution. I focused on ERVs. And then I gave a second presentation, the scientific case or a scientific case for the biblical Adam and Eve, where I touched on all the many genetic reasons why special creation is true and has to be true, as it provides the best explanation, the most parsimonious explanations for much of the data that we have. In my second presentation on Adam and Eve, I focused on mitochondrial DNA, the fact that mitochondrial DNA worldwide can be traced right back to a single female ancestor just thousands of years ago, and all Y chromosomal DNA. Every single male Y chromosome on the planet is nearly identical, very low genetic variation. They can all be traced back to a single male common ancestor, roughly 4,500 years ago, Y chromosome Noah. The Y chromosome DNA is nothing like the chimpanzee DNA. The mitochondrial DNA is nothing like the chimpanzee DNA. This demands separate ancestry. This demands special creation. Because if we can debunk the idea that humans and chimpanzees are related, since supposedly chimpanzees are our closest cousin, this is according to the theory of common descent, that humans and chimpanzees can be traced back to a common ancestor between 6 and 9 million years ago. If we can debunk that idea, then that means that humans and strawberries definitely aren't related through common ancestry. So we can stick as close to home as possible. We can stay with humans and chimpanzees, and we can show that both uniparentally inherited DNA compartments, okay, the maternally inherited DNA compartment, which is the mitochondrial DNA, the powerhouse of the cell, and the Y chromosome that's paternally inherited, that's inherited on the father's side. Both of those go back to 2 single common ancestors, our first parents, okay, but specifically with the Y chromosome, Noah, and they're nothing like chimpanzee DNA. As a matter of fact, with the Y chromosome, if you consider overall gene content, size differences in architecture, the Y chromosome of the human is only about 35% the same as your chimpanzee Y chromosome. Okay, so I want to see a sophisticated mathematical model showing how these drastic, these truly profound changes can occur in just roughly 6 million years since the so-called split. Okay. Lorraine, another good question. And why is the XX chromosome 98% similar to chimps? Here is the important point. Okay. We have a lot of what's called agnostic lines of evidence. If I were to say creation's true because the sky is blue or the earth is round, you'd say, well, that's silly because the evolutionary model can also explain why the sky is blue and the earth is round. This is agnostic to the overall origins debate, creation versus evolution, okay? So what we need to look at is what's called differentiating or discriminatory lines of evidence, data points that only one side can explain, or one side can explain a lot better. And so it is the differences that make all the difference. Let me repeat that. It is the differences that make all the difference. Okay, so we got similarities. We got similarities in DNA, similarities in morphology, anatomy, physiology. Yes. The creation model explains similarities due to common design. Human engineers build and design things in homologous patterns. So does God. We see this in the biological world. This also goes for nested hierarchical patterns. But it is the massive differences that only the creation model can explain in the separate ancestry model that the evolutionary model can't explain, because these massive differences cannot be accounted for in the amount of time that is allotted according to the theory of common descent. For example, with the human and chimpanzee lineages since the split. Okay, so it is the differences that make all the difference. These massive differences cannot be explained by the evolutionary model. So it's the differences, not the similarities. The similarities, the evolutionary model just explains through descent with modification. These structures were inherited by a past common ancestor. Descent with modification, they've been modified over time through selection, environment, and other evolutionary pressures, okay? And the creation model would explain homology in both DNA and in morphology as being the result of common design. So both models can account largely for this observation, okay? But it's the differences that only one model can account for, or account for a lot better than the other model. So sure, we have a high degree of similarity with the XX chromosome. Okay, that's okay. It's the differences that make all the difference. How does the evolutionary model explain the differences? But the thing is, even in similar sequences, let's say you take a sequence in the chimpanzee and the human and compare them, there turns out to be massive differences in the way that those genes are regulated, or the way that those genes are expressed, right? You have DNA to RNA to protein, DNA transcribed into RNA translated into protein. And so there's differences not only in that, but in epigenetics, in alternative splicing, in the architecture of these DNA units and sequences, okay, and gene families. And so even in similar sequences, there are differences. And so this demands special creation, this demands separate ancestry. And the evolutionary community simply, at this point, all they have are stories, rescue devices, rescue mechanisms, and not real empirical reasons, okay, for these challenges. And so right there is just a brief explanation for why separate ancestry is the better and superior model. And in my rant there, so I appreciate it, Lorraine, that gave Peter some additional time to hopefully rejoin us, but it looks like he has not, unfortunately. So what we will have to do is turn this into a two-part series. So I hope everybody enjoyed this impromptu dismantling of ERVs and this impromptu presenting of a case for separate ancestry. Okay. So this has been fun, guys. And again, this is now part one of a two-part series, I think, with Dr. Peter Line, How the Human Brain Refutes Human Evolution. And with that, guys, I'll keep you posted in terms of when we are going to finish this presentation and then address audience questions, as I will save those right now for part two. And so those who have donated to the show and those who have sent in super chats, your questions will be engaged and they will be answered. So God bless all. I appreciate you tuning in, and we'll talk soon. Standing for Truth is out.